Today we're going to be talking about gene regulation in both prokaryotes and eukaryotes. Natural selection favored organisms that have the ability to control their gene expression based on their surroundings, so as not to unnecessarily lose energy. For prokaryotes, we're mainly going to focus on the TRP and LAC operons in E. coli.
The TRP operon is a repressible operon, meaning that it is on by default. When tryptophan is present, the TRP operon remains on, so the DNA can be transcribed by RNA polymerase, and later translated into polypeptides that code for tryptophan-creating enzymes. Tryptophan is the TRP repressor protein's co-repressor, so when tryptophan is present, it binds to the inactive repressor, which then changes the repressor protein's shape and activates it. The active repressor protein binds to the operator, which then switches the operon off, and transcription and translation do not occur. Overall, the TRP operon functions to synthesize tryptophan, which is necessary for prokaryotes such as E. coli's survival. Most repressible operons function in anabolic pathways that synthesize large molecules like the TRP operon, which synthesizes tryptophan and is turned off when the cell no longer needs more tryptophan. The LAC operon is an inducible operon, which means that it is off by default. The LAC operon functions to create enzymes that metabolize lactose, so when lactose is absent, the operon is off. The operon repressor protein is activated by default, so it binds to the operator and keeps the operon off. However, when lactose is present, it acts as the repressor protein's inducer by binding to the repressor protein, changing the protein's shape. Once the repressor's shape is changed, it is inactivated and can no longer bind to the operator. Since the operon is no longer being repressed, the RNA polymerase transcribes the LAC genes. The resulting mRNA is translated into enzymes that metabolize lactose. Most inducible operons function in catabolic pathways that degrade large molecules just as the LAC operon is switched on when there's lactose to be metabolized.
E. coli prefers to use glucose as its energy source, but will produce lactase to break down lactose when there's not enough glucose. The number of small organic molecules called cyclic AMP, or CAMP, increases when glucose is rare. This CAMP then binds to CAMP receptor protein, or CRP, which then binds to the CRP binding site on the LAC promoter. CRP is an activator, which is a protein that stimulates RNA transcription of a specific gene. Basically, it's like the LAC operon on steroids. If the glucose level suddenly increases, the CAMP levels decrease, so the CRP doesn't bind to the promoter. Without its activator, RNA transcription still proceeds, but only at a low rate.
Now we're going to move on to eukaryotic gene regulation, which is a lot more complicated than prokaryotic regulation. We're going to mainly focus on transcription factors, mRNA processing, and chromatin structure.
Chromatin is essentially wound-up fiber made of clumps of histones that contains DNA in the nucleus. The way chromatin is packed can affect gene expression. For instance, in histone acetylation, the addition of acetyl groups open up the chromatin, promoting transcription. Alternatively, in DNA methylation, long strands of inactive DNA can methylate themselves, or add methyl groups to certain bases, usually cytosine. Heavily methylated DNA is not expressed because it's so tightly wound up. DNA methylation can be inherited, which is called epigenetic inheritance. However, it can be reversed in some cases.
In order to start transcription factors need to bind to control elements, which are segments of non-coding DNA. There are general transcription factors, which are included in almost all transcriptions and bind to the TATA box, and specific transcription factors, which are specific to its gene. The proximal elements are closer to the promoter, while the distal elements are farther away, also called enhancers. The rate of gene expression is controlled by these factors, whether they be activators or repressors, and other proteins like mediator proteins help initiate transcription as well. If a gene is being expressed, activators come and bind to the enhancers, or the distal control elements. Then, a DNA-bending protein brings the distal elements closer to the promoter, causing the strand to bend, hence the name. Meanwhile, mediator proteins and general transcription factors bind to the promoter. The activators then bind to those proteins and initiate RNA synthesis. Depending on what the body needs at the moment, it will express those genes because external environmental conditions affect internal available activators. Those specific activators determine which genes will be expressed. After transcription, there is another form of gene regulation called alternative RNA splicing, a process by which many proteins can be produced from the same original RNA transcript. It's when regulatory proteins determine what will be treated as an intron, cut out of the mRNA strand, and what will be treated as an exon, kept and expressed. This dictates which genes will be expressed and which will not.